Now that we've taken a look at how the neurotransmitters get sent across the synaptic cleft, we're going to focus on the receptors. And I'm going to give you two general classifications for our ligand-gated and G protein type receptors. And then we're going to focus specifically on a neurotransmitter acetylcholine and the receptors that it uses. Now we've got two main classifications of receptors. We have ionotropic and we have metabotropic. Tropic, that suffix that you see there, actually it's a root word, means to feed. So we're feeding it something. In the case on the left for ionotropic, we're feeding it ions. So when the neurotransmitter, here's this neurotransmitter here in this little hexagonal red shape, when it comes and binds onto this receptor, it's going to, in this case, open the channel and allow all these ions to come flooding through. Notice that the neurotransmitter itself doesn't go through, it's just causing a change on the receptor. So for these ionotropics, these are really fast acting, meaning that it takes less than 50 milliseconds for them to open up and start doing their thing. So ions can either move into the cell like we're seeing here, or depending on the type of receptor, they can also move out of the cell. Basically, they're going from high to low concentration. We call these ligand-gated channels because they're determined by the ligand, that neurotransmitter that's binding down on here. So that's ionotropic. We've also got metabotropic. So now we're feeding it by metabolism, or a bunch of processes all together. This is much slower. This can take more than 100 milliseconds to do to several minutes for these guys to react. And you're going to see why a little bit later, because it's a much more complicated process. In this case, what happens is we're going to have our neurotransmitters here bind onto this receptor protein, but look, nothing's coming through here. What's actually happening is it's causing a change, and down at the bottom of this we have our old G protein friend with its alpha and beta subunits. And those guys are going to dissociate like you learned before. They're going to cause a bunch of changes, and that's going to cause eventually another protein here on that cell membrane to open up or to close, allowing ions to flow in or out. So we're going to look at this process in a lot more detail, but for right now, I just want you to understand the difference between ionotropic, where those ions go directly through the same protein, that transmembrane or integral protein channel, versus metabotropic, which is going to use our G protein. Now, acetylcholine is a type of neurotransmitter that we're going to spend some time on, and it can be either excitatory or inhibitory, depending on the type of receptor and where that receptor is located. If it's excitatory, we're going to see some of this happening in the central nervous system and on some of our autonomic motor neurons, and it's going to be excitatory to all somatic motor neurons. And I'm going to show you a couple of charts of this so you'll see it, some figures to go with it. It can be inhibitory in some autonomic motor neurons. So autonomics can either be excitatory or inhibitory. All somatics are excitatory. And you can see here, here's our main two types of proteins or receptors again. And we called this kind with the ions going straight on through. Remember, those were ionotropic. And this one over here with our G protein, we call this metabotropic. Well, you're going to see that with acetylcholine, we now have a sub-classification of those. So for acetylcholine, you're going to see this word nicotinic receptor. For acetylcholine, this nicotinic receptor is always going to be excitatory. And for acetylcholine, for the G protein one, we call that a muscarinic receptor. And this is the one that's going to be positive or excitatory or inhibitory. And we're going to look at this in a little bit more detail. So if we start with our nicotinic receptors for acetylcholine, we know just by looking at them that they're ionotropic, because when this acetylcholine, and you can see two of them have to sit on here, when it binds, it's going to allow these gates to open, and we're going to have sodium coming in, and there's going to be a little bit of potassium that goes out, but mostly we're going to have a lot of sodium coming in, and that's going to cause this thing to get excited. It's going to depolarize the cell. So nicotinic ACH receptors are always excitatory, always, 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 whether they're in skeletal muscle in that somatic system or in the autonomic ganglia and all somatic motor neurons that use nicotinic receptors, they're always going to be excitatory. So if they're excitatory, we know that they're making EPSPs. So they are depolarizing that cell because there's more sodium, more positive coming inside. The other one we said was muscarinic, and the muscarinic ACH receptors are metabotropic in their classification. I said they're either excitatory or inhibitory, and this is just one little way that it can happen. Remember, we've talked about alpha and beta gamma dissociating. Well, in this case, the beta gamma is going to dissociate and come over here to this transmembrane protein. And in this case, it's going to open it, allowing that potassium to leave. And if the potassium leaves, it's going to make the cell more negative, so it'll cause it to be inhibitory. There's other cases where we're going to see this thing is going to close off, allowing the potassium to stay in, and it'll depolarize more. So that's how it's going to be excitatory or inhibitory. But I'm going to tell you, these beta, gamma, and alpha proteins of the G protein subunits, they work differently, and we'll take a look at that too. So here's them kind of just in another format altogether. We've got our nicotinic receptors on the left here, and you can see it's just a straight ion channel with its two acetylcholines bound there. Then we've got these muscarinic ACH receptors. So here's our G protein unit, and in this case, it's allowing potassium to come out, so it's getting hyperpolarized, inhibited IPSPs. In this case, it's going to slow the heart rate down, because if we're causing it to hyperpolarize, then it's not going to get excited, so you're not going to get that heartbeat going as quickly. In this case over here, we've got the same kind of G protein and acetylcholine stepping on there, but in this case, it's going to cause this channel here to close off. And if it closes off, it's not going to allow potassium to leave, so potassium stays in, it gets more positive. It could also open up a sodium or calcium channel, in which case we're getting a lot more positivity in here, a lot more cations in here. So this thing will depolarize more quickly, cause it to get excited. And in this case, in the smooth muscles of our stomach, different parts of the digestive system, digestive tract, it's going to cause those to contract. So this is the digest part of rest and digest when we see this in the parasympathetic nervous system. Here's just another schematic way of looking at the same thing, and we're going to see this again when we talk about the autonomic nervous system. So if we divide our nervous system into three different categories, we got our sympathetic, our parasympathetic, and somatic. So somatic's going to our skeletal muscle. Then we got sympathetic, fight or flight, parasympathetic, rest or digest. So all of their neurons initially coming out of here are all going to be our nicotinic. So we can see we got nicotinic here, here, and over here. This one is going to be the motor end plate of the skeletal muscle. So they're all sending out acetylcholine, all going to nicotinic receptors. So they're all ionotropic. They're all excitatory. So they're going to cause the next neuron to fire, or they're going to cause this motor end plate to fire its muscle potential. The other type we said we had was muscarinic. We'll see that down here in our parasympathetic fibers. The postganglionic fiber is going to send out acetylcholine. If it's going to muscarinic, this can either excite or inhibit, again, depending on the receptor. I'm going to save this norepinephrine for later when we talk about the sympathetic nervous system and other types of receptors. Here's kind of a third way just to kind of keep the anatomy in your mind. Remember, our sympathetic nervous system is going to be from T1 to about down to L2. We're going to see those preganglionic neurons coming out, going to the paravertebral ganglion over here, making its first synapse, in which case we just said they're all going to be nicotinic. So they're all going to excite this next neuron in line. And some of those are going to go to sweat glands. In this case, we're going to see muscarinic going to different sweat glands. Here we've got some norepinephrine, so this would be other parts of the sympathetic nervous system, which we'll worry about later. You can also go to the adrenal medulla, and then we'll see a little bit later when we talk about epinephrine and norepinephrine, how those secondary postganglionic receptors neurotransmitter works. You can also see here going to skeletal muscle, acetylcholine, nicotinic receptor excites that. Parasympathetic, we're going to have acetylcholine, nicotinic at first, and then muscarinic going to different cardiac muscle, different glands, and smooth muscle. So just a couple different ways of all seeing the same thing. Agonists are drugs that stimulate a receptor. So in this case, we've been calling them nicotinic receptors and muscarinic receptors, but I haven't told you why. It has nothing to do with the ACH, which is what our body uses. But basically, when we were studying those receptors, we found that nicotine consistently activates those, what we now call nicotinic receptors. So they activate the same thing that acetylcholine is able to activate. So those are those ionotropic receptors for acetylcholine. And that's nicotine as in tobacco, same product here. Muscarinic comes from the muscarine plant, which is really a fancy type of mushroom. And those muscarine is able to stimulate muscarinic receptors. So anything that stimulates a receptor is called an agonist. It's going to make it work faster and better. So antagonists are just the opposite of agonists. Antagonists are going to inhibit that receptor. Here we've got this nice innocent looking plant here. It's called Condodendron tomentosum. Try and say that 5 times real quick. I just had to practice it about 10 times. Every year I got to say this over and over. Condodendron tomentosum. Fancy way of saying curare. And curare basically is an antagonist for nicotinic receptors. Curare is what we use in blowguns or dart guns because what that's going to do, if it's an antagonist, it's going to prevent those nicotinic receptors from working. And we know that nicotinic receptors go to skeletal muscle. So if we prevent those receptors from accepting the acetylcholine, skeletal muscle is not going to contract. So it's going to cause paralysis. Another antagonist comes from the plant Atropa belladonna. Looks like a nice pretty little berry here. Our name for that is just going to be atropine. And atropine is going to inhibit muscarinic receptors. So we're going to see in a little bit that atropine, if we just talked about the heart, and we know that if the beta gamma complex is going to cause an IPSP in the heart, causing that thing to slow down, well, if we have an antagonist for that, so if we antagonize the IPSP, it's going to not slow it down. It's going to allow it to speed up. And that's what we see here with atropine. So this is heart rate in beats per minute. And when we give somebody atropine, it's going to speed up that heart rate because it's not allowing that IPSP to form. Propranolol, which you may have heard, does just the opposite. It's going to slow it down. So that would be like a EPSP, right? It's going to cause that thing to fire off more and cause it to slow down. So here's a kind of the schematic just to put all those together. We've got ACH going to our nicotinic receptors and muscarinic receptors. So it's going to stimulate both of those. So does nicotine for the nicotinic receptors and muscarine for the muscarinic receptors. And then we've got our antagonists, curare for nicotine or nicotinic receptors and atropine for muscarinic receptors. So let's kind of put this all together here. We've got these two types of receptors. We've got nicotinic, which are ligand-gated channels, because remember, it's the ligand that's coming on here, and when it binds, it's opening or closing that gate directly. So those nicotinic for ACH requires 2 acetylcholine. It's not going to do it with just one. It's got to have both of them on there. And again, we said more sodium flows in than potassium comes out. And then we've got our G proteins, coupled channels, which are muscarinic, only requires 1 acetylcholine. And it's going to cause not an action here, but it's going to cause a distant channel to either open or close. I'm going to show you one more slide that's going to kind of detail this whole thing together. So don't let it blow you away when you first see it, but we're going to walk through it slowly. So let's take a look at this slide here. We've got a direct comparison between our ionotropic receptors and our metabotropic receptors, which are going to have 2 different ways of affecting the cell. So if we start with our ionotropic, remember for acetylcholine, we know that the special type of receptors are always going to be nicotinic. And those nicotinic receptors require 2 acetylcholines, and the ions that come through here are going to be excitatory. So this happens really quickly. It's just a one-step process. Boom, you get the 2 acetylcholines, everything comes flooding through here. In the previous picture, we saw some potassium leaking out, but you're going to get a lot more sodium coming into this cell, and so we get an overall effect of excitation. That's just for acetylcholine. Remember, there are other types of receptors that are ionotropic. We're only talking about one here. So some of those other ionotropic receptors could be inhibitory if they were allowing negative ions in, or if they were allowing positive ions out. If we move on to the metabotropic, now this is a little bit more complicated, but you've kind of seen this picture before. If we start off with, now we only need 1 acetylcholine to come in and hit that receptor. And when it does, it's going to activate our G protein here. So if we start, remember there's 2 ways. We've either got the alpha unit or the beta gamma unit. The alpha unit starts off with a GDP, guanine diphosphate, but when this thing gets excited, it's going to kick off that GDP, and it's going to allow new GTP to come on board with the alpha unit. That alpha unit then is going to go over to adenylate cyclase. It's going to use that GDP or GTP, sorry, triphosphate. It's going to use that to activate our cyclic AMP, and that cyclic AMP is then going to go out. We see we're making a bunch of cyclic AMPs. All of those are going to go out and go to phosphate kinases or phosphorylate kinase groups, and those are going to go phosphorylate different molecules or proteins. In this case, what we're going to see is they're going to go out and they're going to activate a protein channel, in this case allowing potassium to leave the cell. So if the potassium leaves the cell, it's going to actually inhibit this cell and cause it to not fire. We're going to lower that membrane potential. So that's one way it can happen. The other way is that the beta gamma subunit can just go directly to another protein channel and activate that one. And it may either open or close those channels depending on the scenario in the cell. In this case, it's opening this channel, so it's allowing, again, potassium to leave, which is going to inhibit the cell. And I just want to clarify, in both of these cases, we're losing positive ions, so we're inhibiting the cell, but this is an amplification of the signal. So the signal is telling it to do something. If it's amplifying an inhibitory signal, or if these channels were allowing sodium to come in or calcium to come in, they would be amplifying an excitatory signal. So keep those 2 in mind. This is going to take a lot longer because of either the alpha subunit is going to have to go through all of these steps, all of these different cascades to get to a bunch of them, or the beta gamma unit is going to have to separately go to a bunch of channels to charge those and open or close those. So a lot of time takes up to go there, but you're going to get a much stronger effect as far as getting more channels open altogether. In the case of the ionotropic, that's the only channel that's opening right there. Now, to give you kind of a couple of examples, with adenyl cyclase, so this ion channel, as we come over here, if we were to consider the stomach, if you were to, in the stomach, this may close. And if you close this channel, then the potassium isn't going to leak out. And therefore, it's going to become more positive because the potassium is staying in the cell. And if it's becoming more positive, it's going to cause those cells possibly to contract. For the beta gamma unit here, if we're allowing this one to open, that's going to create an IPSP. We said it's inhibitory, postsynaptic potential. And that's what we're going to see a lot in the heart with our parasympathetic nervous system. If you had this going on in the heart, you're going to decrease the heart rate because you're reducing the membrane potential by allowing that positive ion to leave. So those are a couple examples, and we'll wrap up this section on the next slide. So this just puts everything in words for you so you can see the words if you like that better. It's the step process of activating and deactivating that G protein. And just to kind of really summarize this, remember when that membrane receptor is not bound, we're going to have this alpha, beta, and gamma complex forming that G protein. And the alpha is going to be holding on to its GDP left over from its prior reaction. But when the ligand comes onto it and binds to that regulatory protein molecule, then the alpha subunit is going to let go of that GDP and bind a new GTP. And this is going to allow the alpha to separate from the beta gamma complex. And then either the alpha goes over to adenylyl cyclase and starts the whole cyclic AMP thing, or the beta gamma complex is going to go to another ion channel. And they're both going to either stimulate or open or close that channel, depending on that channel and where it is. Once they're done, that alpha subunit is going to hydrolyze GDP or GTP, get rid of one of those phosphates, turn it into GDP, which will allow it to come back to that receptor protein and cozy up again next to the beta gamma complex. And the beta gamma complex will come over when it's done with its thing too. So that wraps up these different types of proteins for acetylcholine. Next, we're going to look at another way that acetylcholine is managed in that cleft by an enzyme called acetylcholine esterase.